Welcome to This Week in Gastroenterology. This week we're covering 10 notable papers spanning advances in endoscopy, new pharmacotherapies for metabolic and liver disease, and novel insights into the pathophysiology of inflammatory and genetic GI conditions. Let's dive in. We'll begin with metabolic and liver disease, where new therapies for obesity-related conditions continue to make headlines, and a global study highlights a critical comorbidity in cirrhosis. In Nature Medicine, a Phase 3 trial called SYNCHRONIZE-MASLD evaluated survodutide, a dual glucagon and GLP-1 receptor agonist, for adults with obesity and at-risk metabolic dysfunction-associated steatotic liver disease, or MASLD. This 48-week randomized trial found that survodutide was statistically and clinically superior to placebo on both co-primary endpoints. Over 84% of patients on survodutide achieved at least a 30% reduction in liver fat content as measured by MRI-PDFF, compared to just 24% of those on placebo. Alongside this, patients on survodutide achieved a mean body weight reduction of over 12% versus only 1% with placebo. As expected with this class of drugs, gastrointestinal adverse events were the most common, typically occurring during dose escalation. This adds another powerful agent to the armamentarium for MASLD and obesity, demonstrating significant dual benefit for both the liver and weight. Building on the theme of incretin-based therapies, a study in gastroenterology explored why patient responses to these drugs can be so variable. Researchers identified a distinct subphenotype of obesity, found in about a quarter of their cohort, characterized by fast gastric emptying, increased postprandial hunger, and paradoxically low postprandial GLP-1 levels. This low GLP-1 was linked to reduced mucosal gene expression for GLP-1 synthesis. When they retrospectively looked at patients treated with tirzepatide, this specific subphenotype with low GLP-1 production had a dramatically enhanced response, achieving a 21.5% weight loss at 6 months compared to just 11.7% in other obesity phenotypes. This work is a fascinating step toward personalized obesity medicine, suggesting that phenotyping patients, perhaps by gastric emptying or hormonal response, could help predict who will benefit most from drugs like tirzepatide. Finally, in this section, a large global study in gut puts a number on a common clinical challenge: chronic kidney disease in patients with cirrhosis. This prospective registry enrolled over 7,000 non-electively admitted cirrhosis patients from 127 sites worldwide. The global prevalence of CKD was found to be 18%, with the highest rates in high-income countries, which paralleled a higher prevalence of metabolic syndrome. Patients with CKD had a more complicated course, with more ascites and a much higher risk of developing acute kidney injury during their hospital stay. A staggering 59% compared to 27% in those without CKD. Unsurprisingly, the presence of CKD was associated with significantly higher in-hospital and 30-day post-discharge mortality. This study underscores the prognostic importance of renal function and suggests that meticulous management of ascites and metabolic comorbidities is critical to improving outcomes in this vulnerable population. Next, we turn to the endoscopy suite, with 3 papers looking at improving adenoma detection, managing variceal bleeding, and handling a common ERCP complication. A major real-world study in gastroenterology addressed the ongoing debate about computer-aided detection, or CAID, in colonoscopy. This pragmatic, cluster-randomized quality improvement study was conducted across the United States Veterans Health Administration system. Facilities were randomized to receive CAID devices, and their performance was compared to control sites. The results were clear. At facilities where CAID was available, the adenoma detection rate increased by an absolute 4.2 percentage points, while it slightly decreased at control sites. In a multivariable analysis, CAID availability was associated with a 22% increased odds of detecting an adenoma. Importantly, this benefit was seen across all endoscopist baseline ADR quintiles, meaning it helped both high and low performers. This large, pragmatic trial provides strong evidence that simply making CAID available improves adenoma detection in a real-world setting. 2 studies from gastrointestinal endoscopy highlight technical refinements for therapeutic procedures. The first was a single-center randomized trial comparing two EUS-guided strategies for treating gastric varices. Patients received either coil embolization plus an absorbable gelatin sponge or coil embolization plus cyanoacrylate glue. While both methods achieved 100% technical success and similar rates of initial clinical success, the follow-up data revealed a key difference. The group receiving the gelatin sponge required significantly fewer re-interventions, just 17% compared to nearly 43% in the glue group. The gelatin sponge group also had a significantly lower cumulative incidence of rebleeding. This suggests that for EUS-guided variceal management, the combination of coils with a gelatin sponge may provide a more durable and safer result than the more traditional coil and glue approach. The second paper from the journal addressed oozing-type bleeding immediately following endoscopic sphincterotomy during ERCP. This multi-center non-inferiority trial from Japan randomized patients to either a first-line strategy of applying a self-assembling peptide, or SAP, versus conventional balloon tamponade. The SAP strategy was found to be non-inferior. More compellingly, a pre-specified exploratory analysis for superiority found that the peptide was actually better, achieving initial hemostasis in 92% of cases versus 77% for balloon tamponade. The SAP procedure was also faster and required fewer rescue devices. For this common ERCP adverse event, reaching for a self-assembling peptide first may be a more effective and practical initial strategy. Finally, we'll cover new insights into pathophysiology and treatment strategies for inflammatory and genetic GI diseases. Starting with basic science that has major clinical implications, a report in Nature identifies a novel subset of regulatory CD8 T cells that play a critical role in controlling intestinal inflammation. These cells are guided to the colon by a receptor called GPR15, where they function to kill inflammatory macrophages. The researchers connected this to human disease, showing that deleterious gene variants in GPR15 are associated with severe early-onset IBD, and that these protective cells are reduced in the mucosa of patients with sporadic IBD. This work pinpoints a new cellular player in gut immune homeostasis and identifies GPR15 as a potential therapeutic target for restoring immune regulation in IBD. Shifting to treatment in IBD. A post-hoc analysis in clinical gastroenterology and hepatology provides much-needed data on the speed of onset for advanced therapies in ulcerative colitis. Using individual patient data from 11 randomized trials in biologic-naive patients, the study compared how quickly different drugs provided symptom relief. The primary outcome was a patient-reported outcome response at week 2. Upadacitinib was the clear frontrunner, with 65% of patients responding by week 2, followed by infliximab at about 37%. Compared to infliximab, upadacitinib had more than 3.5 times the odds of achieving this early response. At the end of induction, infliximab demonstrated the highest clinical response rates. This provides valuable practical information for shared decision-making, indicating that for patients needing rapid symptom control, upadacitinib and infliximab appear to be the fastest-acting options. In hepatology, a study in the Journal of Hepatology investigated the mechanisms of T-cell dysfunction in chronic hepatitis B. By analyzing virus-specific T-cells from chronic patients and comparing them to those who achieved a functional cure, researchers defined a 101-gene resolution signature. They discovered that even after HB's ag loss, a transcriptional scar of dysfunction can remain in these cells. Critically, they showed that this dysregulated gene transcription could be targeted. Using an HDAC inhibitor called entinostat in the lab, they were able to improve histone acetylation and restore T-cell function. This provides a strong rationale for exploring HDAC inhibitors as a component of future immunotherapeutic strategies aimed at achieving a functional cure for chronic hepatitis B. We'll close with a paper in Gastroenterology that offers new molecular understanding of sporadic Hirschsprung disease. The study identified a dominant subgroup of patients where key neurogenesis programs were suppressed. This was linked to a deficiency in a protein called PRDM9, which led to DNA double-strand breaks and mosaic deletions in the promoter regions of crucial neurogenesis genes, ultimately impairing the differentiation of enteric neurons. Perhaps most importantly, This process creates a molecular signature of mosaic promoter deletions that can be detected in blood. A blood-based score combining this signature with a polygenic risk score was able to discriminate Hirschsprung cases from controls with an impressive accuracy, achieving an AUROC of about 0.9. This work not only provides a new mechanistic explanation for many sporadic cases, but also opens the door to a non-invasive, blood-based molecular test to help stratify patients. If you only have time for one paper this week, make it the study on computer-aided detection in colonoscopy from Gastroenterology. This large, pragmatic trial from the United States Veterans Health Administration system provides some of the best real-world evidence to date that making CAID technology available in endoscopy units significantly boosts adenoma detection rates, a key quality metric linked to cancer prevention. Here are the key takeaways from this week in gastroenterology. First, in the real world, the availability of computer-aided detection for colonoscopy significantly increases adenoma detection rates across all levels of endoscopist performance, supporting its wider adoption. Second, for patients with moderate to severe, biologic-naive ulcerative colitis needing rapid symptom control, upadacitinib and infliximab demonstrate the fastest onset of action, with superior response rates at week 2. Third, in metabolic disease, survodutide shows potent dual efficacy for both liver fat reduction and weight loss in MASLD, while a newly identified obesity subphenotype with low GLP-1 production shows a particularly strong response to tirzepatide, hinting at future personalized approaches. Fourth, for endoscopic management of gastric varices, EUS-guided coil embolization combined with gelatin sponge may be superior to coil plus glue, as it significantly reduces the need for reintervention and has a better rebleeding profile. And finally, chronic kidney disease is present in nearly 1 in 5 hospitalized patients with cirrhosis globally and dramatically increases the risk of in-hospital AKI and mortality, highlighting the importance of renal management in this population. That's your roundup for this week in gastroenterology. The full transcript and references are available on the episode page in your Audio Scholar library. This is an AI-curated summary. For clinical decisions, always consult primary sources and current guidelines. See you next week. This is an automated summary generated by artificial intelligence, which can make mistakes. Always review the original source materials.